Good afternoon, morning, evening, whenever you happen to be watching this, everybody. What I'm going to do today is kind of take you through the first couple stages of cell respiration here. And as I do that here, I'm realizing, there we go. As I do that here, we're going to try and make sure that we hit all the highlights and all the key points here. And then if you have any questions about anything that I go through in these videos, please sign up for an academic support time. So that I can help you out with your questions that you have. I'll also give an opportunity to ask questions in class, but I know sometimes people would rather have that one-on-one time to ask questions. So please, please, please sign up for that academic support time if you have any. Okay, so what we're going to be looking at here is cell respiration. Remember, the point of cell respiration in the end is to make ATP, right? So the beginning stages kind of set the tone, set the stage for making the ATP in the end. Okay, so what we're going to be doing in cell respiration is harvesting the energy that is stored in our organic molecules that we eat, in the carbohydrates, in the fats, in the proteins, even the nucleic acids that we eat, right? Now, heterotrophs, we eat these organic molecules, right, as our food source. So squirrels, humans, lions, right, even the dead zebra there would eat grass or whatever else is available. And then we digest those organic molecules in order to get the raw materials we need for synthesizing our own molecules, right? When we eat protein, when we eat, say, like a burger, right, that's cow protein that we're eating. And then our body will digest those cow proteins into individual amino acids that we then use to make our own human proteins, which are different than cow proteins. The food that we eat also serves as the fuel that we need for energy, and we control the release of the energy in those molecules. Those food molecules that we eat, store a whole lot of energy, and if we released all that energy at once, it would be the increase in temperature would be too drastic for our cells to deal with it. Okay, so instead, we slowly release the energy in these molecules in a bunch of small little steps, and each one of those steps is controlled by enzymes. Okay, so the model molecule that we're going to take a look at as we study this is going to be glucose. Keep in mind, any food that you eat can enter into cell respiration. They don't always enter at the same part of the pathway. Some of them enter a much later than glucose would into this biochemical pathway, but all the foods that you eat, in order to extract energy from them, have to go through cell respiration. We're going to focus on glucose. We'll take a brief look at the end of our cell respiration lectures on the ways that some of the other nutrients that we eat pass through cellular respiration. So we're going to break down glucose. It's going to be catabolized in order to produce ATP. So here is our equation for cellular respiration. We're going to take glucose and oxygen, and then we're going to break it down into water and carbon dioxide plus energy that gets released. That energy is in the form of ATP, although there is some heat energy generated also that is just lost, that is not used for doing any work. Okay, so this is basically a combustion reaction, right? Because you're taking an organic molecule and burning it, breaking it apart in the presence of oxygen, and water and carbon dioxide are your products. So hopefully you remember a little bit of your different reaction classifications from chemistry, right? This is a combustion reaction. Now, in real life, combustion, out in your driveway or in your backyard, you might have a fire pit, right? And again, your fuel is going to be carbohydrates. You're going to add some heat in the presence of oxygen. You're going to combust that wood, and it releases carbon dioxide and water and heat energy. Okay, so in cellular respiration, we're going to make ATP and some heat energy by burning fuels in many small steps, whereas when you burn that wood, it's one big step, right? We're going to break this down into many, many small steps using enzymes, right? So how do we harvest the energy from these foods, right? We're going to break these large molecules into smaller ones. And then, this is the key, we're going to move electrons around from one molecule to another. And in general, the way these electrons are moved is they are moved from a less electronegative molecule or a less electronegative atom within a molecule to a more electronegative molecule. Okay, and what that does is it causes that electron, every time it moves to a more electronegative molecule, it is moving to a more and more stable configuration. And as anything moves from a less stable configuration to a more stable configuration, it releases energy. Okay. So as these electrons move, they are going to carry energy with them. And that energy is going to be stored in another bond. It could be released as heat, or it could be used to make ATP, right? So what we're doing is moving electrons from one molecule to another, or from one atom to another. So one molecule is going to lose electrons and the other one is going to gain electrons. Okay. Now this is a special kind of reaction in chemistry. This is called a redox reaction, or an oxidation reduction reaction. Whatever atom or molecule loses electrons is said to be oxidized, and whichever one gains electrons is said to be reduced. So what we're going to be doing or what our bodies do in cell respiration is remove electrons from glucose and pass them to other molecules. So in the process of cellular respiration, glucose is being oxidized. Okay. Now it might sound weird that the atom that gains electrons is said to be reduced, but remember, we're talking about the electrical charge here. So the electrical charge is becoming more negative, or is getting reduced in the atom or molecule that gains electrons. Okay, so when we move electrons in living things, we can't just strip the electrons right off of these atoms. The electrons are going to be moved as part of a hydrogen atom, right? So we're going to remove hydrogen atoms from our molecules during cellular respiration and transfer them to other places. Okay, now those hydrogen atoms don't just like fall off of glucose. They're removed by special enzymes called dehydrogenases, and there's lots of different dehydrogenases that remove hydrogen atoms during the course of cellular respiration. I'm not going to expect you to memorize the names of any enzymes other than one that we'll take a look at during cellular respiration. That's because it's a very important control enzyme in the process of cellular respiration. All the rest, I don't expect you to know, but I would expect you to know that dehydrogenases remove hydrogen atoms and move them to other molecules. And of course, I expect you to know that kinases in general are going to phosphorylate molecules, right? They're going to transfer phosphate groups onto other molecules. So when we move a hydrogen atom from one molecule to another, we're taking its electron with it and moving that. Okay, so moving hydrogens in cellular respiration is moving electrons. Okay, so as we remove a hydrogen atom from one molecule, it loses electrons and is oxidized, and we transfer it to another one. That molecule that gains those hydrogen atoms is reduced. Okay, so here glucose is converted to carbon dioxide, so it is being oxidized, right? Water is converted, oxygen is converted into water, and so the oxygen is said to be reduced. Okay. All right, so just to make sure that we're clear here, this is just kind of a generalized reaction here. And we've got a molecule X that has an electron on it, and molecule Y does not currently have that electron, as it gets transferred in this reaction, this would be a redox reaction, right? Molecule X is losing an electron and molecule Y is gaining an electron. The molecule that donates the electron is said to be the reducing agent. It is what causes the other molecule to be reduced here. So X is the reducing agent. The molecule that receives the electron is said to be the oxidizing agent, because it has caused the molecule X in this case to become oxidized. Okay. Now, when we look within what's going on in cellular respiration, a lot of times what happens with these redox reactions is simply the degree of sharing of these electrons changes. Okay. So for example, when we look at the combustion of methane, which is CH4 in the presence of oxygen, CH4 is converted into carbon dioxide. So it's losing these hydrogen atoms. It is being oxidized. Whereas water is gaining these hydrogen atoms, or oxygen, I'm sorry, is gaining these hydrogen atoms and being converted into water. And so it is being reduced. Now, if we take a look at what happens here, okay, our oxygen atom, or our oxygen molecule here, shares those electrons equally, right? But in the product water, we know that oxygen has a much higher electronegativity than hydrogen does. So those electrons end up much closer to oxygen. This is much more stable for these electrons to be closer to this oxygen atom than to be equally shared between oxygen atoms. So we went from a less stable configuration for those electrons to a more stable configuration. And so energy is lost there during the course of this reaction. Okay. Oxygen is highly electronegative, so it is a very potent oxidizing agent. And in fact, because it is such a powerful oxidizing agent, that's why this is called oxidation when an atom or molecule loses electrons, because it's usually losing them to oxygen. Okay, so these electrons are losing their stored potential energy when they shift from a less electronegative atom towards a closer to a more electronegative atom, because they're moving from less stable to more stable. And so in that case, then energy gets released. All right. So what we see in cellular respiration is many, many, many redox reactions that occur. And each one of those redox reactions leads to a little bit of energy being released as those organic molecules are being oxidized and reduced. Okay, so during cellular respiration, there's going to be carbon-carbon bonds that are broken. And then we're going to remove electrons from carbon-hydrogen bonds by removing hydrogen atoms. So in this case, what happens in cell respiration is our fuel, our glucose, which is the fuel, is oxidized as it's converted from glucose into carbon dioxide. It's lost all those hydrogen atoms with their electrons, so it has been oxidized. Those electrons are attracted to more electronegative atoms, and in biology, the most electronegative atom that we know is oxygen gas, okay? So in this case, when oxygen is converted into water, the oxygen has been reduced. It has picked up those hydrogen atoms with their electrons. Oxygen has gained electrons in this reaction, so it has been reduced. So within our cells, we're going to couple these redox reactions together, and we're going to use the energy that is released by those redox reactions to make ATP. Because remember, adding a phosphate group onto ADP is an endergonic process. It requires an input of energy, and that energy comes from all the energy that's released in these many redox reactions that occur. Okay, so just to kind of summarize the difference between oxidation and reduction, oxidation is removing hydrogen atoms, therefore electrons, when we're talking at cellular respiration, and reduction is adding hydrogen atoms and thus adding electrons to an atom or molecule. So oxidation is the loss of electrons, reduction is the gaining of electrons. Oxidation reactions release energy, and then reduction reactions store energy, okay? So oxidation reactions are exergonic as these electrons are being stripped away, energy is being released, and reduction reactions are endergonic, okay? Now, in cellular respiration, there are very specific important carrier molecules that move and shuttle these hydrogen atoms around inside of the cell and inside of the mitochondria, okay? And these electron carriers are made from vitamins that we would eat, like vitamin B3, which is niacin. And the electron carrier molecule that is most commonly used in cellular respiration is called NAD+, nicotinamide adenine dinucleotide, which is why we call it NAD. Because it's just a really long name to say. Now, when it has not picked up any hydrogen atoms and is not shuttling hydrogen atoms around, it is in this NAD+ state, which is its oxidized state. When it picks up a hydrogen atom, it is reduced and becomes NADH, and that's when it is shuttling electrons around. Another less commonly used, but still sometimes used electron carrier in the cell that is kind of a very close cousin of NAD+, is FAD+2, which is flavin adenine dinucleotide. And it also picks up hydrogen atoms and becomes reduced when it picks up those hydrogen atoms, becomes FADH2, okay? All right, so when we add hydrogen atoms here, our NAD+ is going to be reduced. We're gonna add that hydrogen atom right here to this ring in the NAD+, and it becomes NADH. And so this NADH is now shuttling electrons around within the cell.
Oink, oink.
Okay, and so that is stored energy that can be used to reduce other molecules, and is energy that is stored that can be tapped into later, like money in the bank. Okay, so when we talk about cellular respiration, there's 4 basic stages to cellular respiration. Today, I'm going to be going through glycolysis and probably the Krebs cycle. Okay, so there's an anaerobic stage that does not require oxygen to be present, and that first stage is called glycolysis. It occurs without oxygen, and it occurs in the cytosol, not within the mitochondria. In the cytosol is where this takes place. Then there is the aerobic stage of cellular respiration, which occurs in the presence of oxygen. Oxygen must be present for this stage, for these stages to take place. These occur in the mitochondria, okay? There's what's called pyruvate oxidation, where we're going to take the final product of glycolysis and we're going to oxidize it. There's a stage called the Krebs cycle. It's also called the citric acid cycle in some places, and also the tricarboxylic acid cycle in some places. So if you see any of those names, Krebs cycle, citric acid cycle, tricarboxylic acid cycle, those are all the same thing. Okay, and then the last stage is the electron transport chain. Okay, so remember what the point here of all of this is to make ATP. So let's dive into the first stage. Let's dive into glycolysis here. So let me pull this up and share this with you. So we're going to take a look now at glycolysis, the first stage of cellular respiration. All right. Remember, we're trying to make ATP here. So we're going to break down glucose in glycolysis. If we take a look at the origin of this word, glyco means sugar, lysis means splitting or dissolving. So we are splitting sugar here during glycolysis. And what is going to happen is that 6-carbon glucose molecule is going to be split apart into 2 3-carbon molecules called pyruvate or pyruvic acid. You'll see both of those terms. So this is a really ancient pathway. This is one of the very first biochemical pathways that evolved to provide living things with energy. So it's where energy transfer first evolved. We're going to transfer energy from our organic molecules like glucose and store it in ATP. This glycolysis is the starting point for all cellular respiration in all organisms. However, it's very, very inefficient. We only generate 2 ATP for every 1 glucose. That means we tap into about 3.5% of the stored energy in glucose to make 2 ATP. So this is highly inefficient, a highly inefficient process. Okay, for multicellular eukaryotes like you and I, 2 ATP for every 1 glucose is not going to provide us enough fuel to survive. And remember, this occurs in the cytosol. Okay, now this makes sense evolutionarily because if we think about this from an evolutionary perspective, this process first evolved in prokaryotes. And we know, what do prokaryotes not have? Organelles, right? They don't have membrane-bound organelles. They don't have a nucleus, but they don't have mitochondria, for example, or Golgi bodies, or endoplasmic reticulum. They have ribosomes, but no membrane-bound organelles. There's no membrane around ribosomes there are around all those other organelles. So prokaryotes were those cells with no organelles. Early Earth's atmosphere was an anaerobic atmosphere. Very little oxygen was present, right? So life on Earth first evolved about 3.6 billion years ago or so without any free oxygen, any O2 in the atmosphere. Free oxygen means oxygen that is not tied up in another molecule. Like the oxygen in glucose is not free oxygen because it's tied up with all those carbons and hydrogens, right? Free oxygen just means it is O2, right? It's free in the atmosphere. So the energy that was found in those organic molecules that these prokaryotes were taking in had to be captured without any oxygen being present. Okay, so some prokaryotes evolved the process of glycolysis, and those would be the ancestors of all modern life. All cells today still use glycolysis. Okay, so what we say is that the enzyme, and this is another piece of evidence for a common ancestor, is the enzymes of glycolysis are well conserved. And when we say well conserved, that means they really haven't changed very much over time. They are still very much the same that they were 3.56 billion years ago. So no, you do not have to have these bacteria over for the holidays. I would recommend that you don't have many of them over for the holidays, especially this year. although it's not a bacteria we're worried about this year, it's a virus, of course. Okay, so glycolysis occurs in 10 steps, and it converts glucose to those 2 3-carbon molecules called pyruvate. Okay, the first stages of glycolysis, the first 5 stages, are referred to as the energy investment phase. The cell is going to spend a little ATP, use it at the very beginning of glycolysis to kind of get this process started. What it's going to do is it's going to transfer phosphate groups from ATP onto glucose in order to destabilize the molecule so it's easier to break it apart. Of course, what kind of enzymes are going to be needed to transfer phosphate groups from ATP onto our organic compounds in cellular respiration? Right, kinases, okay? So we're going to convert glucose to those 2 pyruvic acid molecules, okay? After the energy investment phase, we're going to get what's called the energy payoff phase, where we're going to actually remove some hydrogens from our organic molecules that are our intermediate molecules between glucose and pyruvate. We're going to start oxidizing some of our organic molecules and reducing NAD+ to generate NADH. Okay, we're also going to generate ATP. 4 ATP are going to come out at the end of glycolysis. Remember, we spent 2 at the beginning, so we're going to get a net gain of 2 ATP. All right, so we get a total of 4 ATP and 2 NADH produced. Remember, we use 2 ATP at the beginning, so we get a net gain or net yield of 2 ATP and 2 NADH in the end. Okay, now, of course, all these steps are catalyzed by enzymes, right? None of these steps occurs in a vacuum or all by itself or spontaneously. It would be naive to think these happen spontaneously. So like I said, the first stages are endergonic. There's the energy investment phase where we use a little bit of ATP to get this process started, right? And that glucose is eventually going to be destabilized, and it breaks apart into 2 3-carbon molecules. One is called DHAP, the other is called G3P. And it is the G3P that continues on through the rest of the stages of glycolysis. And there's an enzyme that converts the DHAP, DHAP, dihydroxyacetone phosphate 3-carbon compound into G3P so that can also go through the rest of the stages of glycolysis. So we have then the energy payoff phase, which is like the last 5 stages of glycolysis or 5 reactions, and those are exergonic. A little bit of ATP is produced and a little bit of NADH is produced. And remember that NADH has these a bunch of these electrons whose energy we can harvest later to help us make ATP. Okay, so we gain net gain of 2 ATP and 2 NADH. Okay, so here are all the stages of glycolysis, and notice each stage is catalyzed by a different enzyme. Okay, you do not need to know the names of all of these enzymes. You do not need to know the names of all of these intermediate molecules. You should know G3P, glyceraldehyde 3-phosphate. This molecule here is what proceeds through the rest of cellular respiration. And we're going to see this molecule again in photosynthesis. So you should be familiar with G3P. You should be familiar with this enzyme right here, phosphofructokinase. This is my favorite enzyme. I have a second favorite, but this one is my first favorite enzyme, phosphofructokinase. We're going to talk about what it does and why it's really important when we talk about how cell respiration is controlled at the end of our cell respiration lectures. Okay, but you can see some other types of enzymes that you should recognize: kinase, dehydrogenase, right? So you should be able to just see the name and go, oh, it's going to add phosphate groups onto our molecules. And look at this, sure enough, it is. Dehydrogenase, oh, that's got to be removing hydrogen atoms. And sure enough, it must be, because we're adding hydrogen atoms onto NADH. Those must be coming off of glyceraldehyde 3-phosphate. Okay, so this first half of glycolysis, these first 5 reactions, the addition of these phosphate groups serves to make these organic compounds that are here much less stable and much more reactive, so they will split much easier. Okay, and so this destabilized glucose that is now in the form of fructose 1,6-bisphosphate, which has a lot more stored potential energy than glucose does because it has these additional phosphate groups on it, is very unstable, and it will split And it splits, like I said, into this DHAP, dihydroxyacetone phosphate, and glyceraldehyde 3-phosphate. But there's an enzyme that converts that DHAP into G3P, which then goes through the rest of the stages of cell respiration or glycolysis. Okay. So, like I said, that phosphofructokinase is key and G3P is key. Now, the second half of glycolysis is where we get our energy produced finally. Okay, we get some NADH generated right there. Okay, and remember that we saw on the previous slide that was done by a dehydrogenase. Okay, so G3P is donating hydrogen atoms with their electrons to the NAD plus molecules, reducing them and creating NADH. Okay, that oxidizes that G3P molecule because it is losing electrons when it loses those hydrogen atoms. And like I said, it reduces the NADH. So we have a redox reaction here, right? The sugar is oxidized, the NAD plus is reduced to NADH. We get a little bit of ATP generated, right? We get some ATP generated here at this stage, and we also get it generated down here, okay? So what we have here with this ATP production is that we get a sugar called PEP, phosphoenol pyruvate, that has got a phosphate group on it that is going to be transferred from the molecule directly to ADP to generate ATP, okay? Because the phosphate group is coming off of this PEP molecule, this is called substrate level phosphorylation. We're transferring the phosphate group from a substrate, from an organic molecule, directly to ADP to generate ATP, okay? We're going to see a different type of phosphorylation occurs in the last stage of cellular respiration in the electron transport chain. We're going to see what's called oxidative phosphorylation occur. And you need to know the difference between the two. So right now, you need to make sure you're paying attention to this substrate level phosphorylation that produces ATP from ADP. Okay, so in the last steps of glycolysis, where did the phosphate come from to make ATP? It came right off of the sugar substrate phosphoenol pyruvate. Okay, and so we have an enzyme here called pyruvate kinase, and in its active site, we're going to have that PEP, phosphoenol pyruvate, with its phosphate group attached to it, bind to our substrate as well as bind to our enzyme as well as ADP. So PEP and ADP are the substrates for this pyruvate kinase enzyme, which in this reaction is going to transfer this phosphate group to ADP to generate ATP, and the molecule that is left behind is called, the 3-carbon molecule left behind is called pyruvate. Okay, and so the phosphate is transferred from PEP to ADP by a kinase enzyme generating ATP. Okay, so the phosphate group came directly from the substrate here, right? And we're going to get 2 pyruvate molecules generated because we started with a 6-carbon molecule called glucose that gets split into 2 3-carbon molecules, DHAP and G3P. And then G3P, we get 2 G3P for every glucose, and we're going to end up with 2 pyruvate for every glucose because they have 3 carbons in them. And to make, we haven't lost any carbons anywhere yet, and so we're going to have, we still have a total of 6 carbons within cellular respiration, just split up between these 2 3-carbon compounds. Okay, so if we take a look at what we get out of glycolysis, right? We invest a little bit of ATP at the start, and we get some out at the end for a net gain of 2 ATP. We also generate some of those electron carriers, the NADH, and the energy stored in those electrons is going to be harvested later in the electron transport chain. So we had a net gain of 2 ATP and 2 NADH, right? So it's a very small energy return. And then we split our 1 6-carbon sugar into 2 3-carbon sugars called pyruvate or pyruvic acid. Okay, so that's not a whole lot of energy, right? But for over 1 billion years of life, this is how life on Earth survived. There was no oxygen in the atmosphere, and so growth was slow, reproduction was slow, because glycolysis, like I said earlier, only harvests about 3.5% of the energy that is stored in glucose. There's a lot more carbons still to strip off and a lot more hydrogens to remove from those carbons by the time we get to the end of glycolysis. So there's a lot more energy to harvest from these molecules. Okay, but without oxygen around, that's all that can be done. Okay. All right. So just kind of showing us one of the issues if you are an organism that only can carry out glycolysis is that eventually your cell is going to run out of its supply of NAD plus to help to oxidize your fuels, right? You won't have anywhere for your cells to donate those hydrogen atoms if these NADHs have nowhere to go, have nowhere to drop off their electrons, okay? So cells that only carry out glycolysis and don't carry those electrons to an electron transport chain inside of a mitochondria have to have somewhere to drop off these electrons from NADH so that the NAD plus gets recycled and can go back to glycolysis to pick up more electrons, okay? Because otherwise energy production would stop and the organism is going to die. So there must be another molecule in these cells that accepts hydrogen from NADH so that the NAD plus is freed up for another round. So how is NADH recycled to NAD plus in cells that only carry out glycolysis? They have to drop those electrons off to another molecule, okay, in order to recycle it. Now, when oxygen is present in aerobic respiration, the NADH goes into the mitochondria, drops off those electrons at the electron transport chain. They eventually end up attached to oxygen and make water, and the NAD plus is recycled and goes back to glycolysis. But without oxygen present in the cell, then cells have to go through what's called fermentation. Some cells, like bacterial cells, can only carry out fermentation. Some of our cells can only carry out aerobic respiration. Our brain cells, for example, never switch to using fermentation. Our muscle cells, though, can switch in low oxygen environments to fermentation for a short period of time, okay? And some organisms in this process of fermentation will either carry out lactic acid fermentation or ethyl alcohol fermentation, okay? These are not to be confused with anaerobic respiration. This is not anaerobic respiration. Aerobic respiration, oxygen is the final electron acceptor in the whole biochemical pathway. In anaerobic respiration, some other molecule is the final electron acceptor, and some organisms like sulfur atoms become the final electron acceptor. So those would be, that would be anaerobic respiration if an organism uses a form of respiration that has something other than oxygen as the final electron acceptor. So it goes through glycolysis and the Krebs cycle and the electron transport chain, just has something other than oxygen as the final electron acceptor. Okay, these lactic acid fermentation and alcohol fermentation are not anaerobic respiration. They are anaerobic processes. They occur without oxygen being present, but they are not considered to be anaerobic respiration. So in some bacteria and yeast, the fermentation that takes place is going to be the ethyl alcohol fermentation, okay? And this is used taken advantage of by humans to generate beer and wine and bread. Okay, and then animals and some fungi and some bacteria are going to carry out lactic acid fermentation. So we carry out lactic acid fermentation. We've been exercising for a long period of time, and our oxygen levels get low. Our muscle cells will switch to fermentation. Okay, and this is used to help make cheese, and it's also used during athletic activities. Okay, so real quick, let's take a look at the alcohol fermentation that occurs in bacteria and yeast. Okay, and what we're going to get here is we're going to start with pyruvate, right? Because that was the final product of glycolysis, and we're going to end up generating ethanol and carbon dioxide through this alcohol fermentation process. Okay, so if we take a look at what happens here, we start with glucose, it goes through glycolysis, it generates pyruvate, right? And in the process of glycolysis, 2 ATP and 2 NADH are generated. But we got to have somewhere for these NADH to drop off their electrons that they've picked up so that NAD plus is regenerated and can go back to glycolysis. So what happens here? The pyruvate that is formed, an enzyme removes CO2 from it. This portion of the molecule right here gets pulled off. And that generates a molecule called acetaldehyde. The NADH drops off its hydrogens and their electrons with acetaldehyde, which converts it into ethanol and converts the NADH back to NAD plus to go back to glycolysis in the cytoplasm. This, however, is a dead-end process because if the ethanol builds up to too high of a concentration, that will kill yeast and bacteria that generate it. And this reaction cannot be reversed. We can't take this ethanol and convert it back into pyruvate. Okay, so that's what happens in some bacteria and yeast. In animals and some fungi and some bacteria, lactic acid fermentation takes place. And with this, we're going to convert pyruvate directly into lactic acid. Okay, now, if we take a look here at this process, we see that glucose goes through glycolysis. We generate the 2 ATP and the 2 NADH and our 2 pyruvate here. But unlike the alcohol fermentation, there's no enzyme that removes this CO2 from our pyruvate molecules. By the way, the CO2 that is produced during alcohol fermentation, that's what causes bread to rise. When you're baking bread and you have yeast in there, those yeast are generating the CO2 gas that causes the bread to rise, and they also generate alcohol, but that evaporates off during the cooking process. Okay, but here in animals and some fungi and some bacteria, we don't pull the CO2 off of this pyruvate. It remains a 3-carbon compound, and the hydrogens with their electrons are transferred directly from NADH onto the pyruvate molecule, and that's what generates the lactic acid or lactate molecule. This is reversible. The lactate can go to your liver and be converted back into pyruvate, which can then finish off the rest of the stages of cell respiration once oxygen is available to the organism. Okay. So this, so once O2 is available, the lactate is converted back to pyruvate by the liver, like I was saying. Okay, so pyruvate is our branching point between fermentation and aerobic respiration. If no oxygen is present or available for the cell, or if oxygen concentrations are low, then the cell will use fermentation. Or if the cell just doesn't have mitochondria or isn't capable of aerobic respiration, then it will carry out fermentation. If oxygen is available to the cell, then that pyruvate molecule will enter the mitochondria, go through the Krebs cycle, and the rest of aerobic respiration. Okay, so remember the point here is to make ATP, and we're going to take a look and see how that is done as we get into the Krebs cycle and the electron transport chain. However, I think that's long enough for this video for today. So that's where I'm going to stop, and hopefully what I said today, what I talked about made some sense. And like I said, if there's things that you're confused about, ask about them in class or sign up for an academic support and see me there. Okay. Have a great day, whatever's left of it for you whenever you're watching this, and I will see you next class.